Hello, everyone. So we're going to switch gears a little bit. So I'm a faculty with University of Missouri Columbia. This work is mainly led by my postdoc there, Yushu Jiang, and several other students. So I just wanted to start with a brief overview of my lab. We really focus more on enabling translational multiomics research, taking large omics data sets, multiomics data integration aspects, whatever is known in terms of functions, pathways, and knowledge that exists in the public domain. And we really work with diverse applications, all the way ranging from plants to animals to biomedical side to viruses. So don't be surprised if you hear everything together in one. In my lab, we do focus on three different aspects. One is the multiomics framework. So we do development of tools for enabling web-based access for a lot of our collaborators and really use large data sets to, predict things and then present to them. You may have seen a couple of posters down, especially the allele catalog work and some multiomics work in kidney. A lot of new tool development, which is really targeted towards application. We really want to bring it back full circle to our collaborators so they can make changes in, apply it in precision medicine, apply it in precision agriculture. You will hear one more talk, GLP Deep, tomorrow in the morning session by one of my students, Sania. And today I'm really going to touch upon this IRnet part, which is work done by my postdoc. Here we are really focusing our work in terms of cancer. And if you look at the cancer statistics just for 2024, you're going to have more than 2 million new cancer cases. And in spite of all the advances in treatments and diagnosis and so forth, the prevalence, the incidences, and then the death rates still are on the rise. I have highlighted a couple here, which are mainly focusing on stomach, bladder, and melanoma here, because that is what you will see me talk about the most. Cancer treatments, there are several different options for patients, ranging all the way from hormone therapy, surgery, chemotherapy, and so forth. For this talk, we are only going to be focusing on things under the immunotherapy aspect. Now, immune checkpoint inhibitors are normal part of our immune system. When the checkpoint and the partnering proteins bind to them, they give a off signal to the T cells, which really prevents the immune system from destroying the cancer. And that's really not what is helpful for the patient. So a class of drugs called immune checkpoint inhibitors, or ICI for shorts, are really aimed at blocking these checkpoint proteins from binding. And they fall under many different categories. But for today's talk, we're going to focus more on the anti-PD-1, PD-L1, anti-CTLA4, in case you're not sort of familiar with those. So what you see here on the left-hand side of the screen is the anti-PD-1 and PD-L1 mechanism, and they are tiny. Hopefully you see them a lot bigger there on the screen. But the PD-L1 and then the PD-1 protein here on the T cell, the upper top one is the tumor cell, they would normally have this binding going on, which inhibits the killing of the tumor cell. But once you have these anti-PD-L1 and PD-1, sort of the drugs that we were just referring to, block these sites, they will really encourage the binding and the killing, which is really what we are hoping to be able to predict if the patient responds to. Anti-CTLA is again very similar, except for this part, CTLA4 binds with BT, B7, 1, and 2 proteins, and they normally would inhibit the T cell activation. Whereas here, when you block the CTLA with the anti-CTLA portion, it's really going to start activating the killing. And so both these mechanisms are really what we are looking for. The motivation for this sort of work is really over the years, ICIs have become the first and the second line of treatment for a variety of cancers, melanoma, gastric, head and neck, and so forth. And the list is big, which is what you'll see for each class of the drug, what type of ICI it is, and what sort of cancer treatments or cancers are targeted for treatment using them. Now, a small percentage of patients do respond to the treatments. However, there are some others who are non-responders or have significant side effects. And this is what we really want to prevent. Knowing ahead of time, it would be good because these treatments are expensive, nor do you want the patients to undergo severe adverse reactions, which can again cost a very large amount. And really, we want to be trying to get some information ahead of time so better choices and alternative treatment strategies can be looked at. So using large amounts of multiomics data is one of the approaches, and several groups have targeted in trying to come up with computational methods, especially Impress, Tide, and NetBio, the ones that we are really going to be comparing our method against. And our goal behind that was because we have seen several limitations for these methods. Some of them are really only focused on using some of these already known biomarkers, so it doesn't really allow you to go beyond that. These biomarkers are often noisy and are biased. Very few methods really take you and provide you options of running and doing these, utilizing these tools all the way to the end. And you really don't want clinicians going to, a Linux server. And again, I think somebody touched upon it, installing conda environments and things like that. So you really want to bring that out of the equation for better utilization. And then prediction accuracy and performance is also very unstable in some of these cases. So that's really what we are targeting with our method here called IRnet. And we really want to predict simplest things. So essentially, whether the patient is going to be a responder or a non-responder. And IRnet utilizes biological information to transfer the genes into the pathway. So we will be using entire transcriptome data, not necessarily only a subset or the biomarker information. We use the gene membership, but in the context of pathways. And then we have this pathway network, which is what we are going to be portraying that on top of it. Now, besides doing the prediction, we really would also be quantifying things and telling the users about pathway information, pathway interactions, and really the relevance of the gene for those purposes. And then the hardcore method part of that really utilizes graph attention neural network layers and global attention pooling layers in between, which are really going to be relevant to individual patient and predicting if they are going to be responders or non-responders. Now, in doing so, of course, we have some challenges. We definitely want to compare our methods with the others. And so we are trying to use similar data sets that they have used. And you'll see here in this table data sets referring to melanoma, gastric, and bladder cancer from the individual publications and the methods, and which is what we are really going to cross compare against. However, some of these do have limitations in form of smaller number of patient samples. Some are imbalanced, which means you have way more number of non-responders versus responders. Also, different cancer types or drug treatment strategies. Some are only PD-1, some are anti-PD-L1, and things like that. So to address some of these limitations, we did incorporate transfer learning to increase the sample size, bootstrap and focal laws to balance the data sets, and then use specific models which can really, be used to counteract the limitations of the databases and the batch effects. And then the validation results indicate that using all of these three strategies, our performance is a lot better as compared to just our baseline. So jumping into the results, by within cohort, I mean whenever the training happened on the same cancer and the testing's happening on the same cancer. So I've put in the red, marked the data sets here for your convenience. Melanoma, and then the vanilla is the baseline, which is without bootstrap or transfer learning. And as you proceed to the columns on the right, you will see that many of the ones that I've highlighted here in the red are where we are gaining, improvements in our performance. So definitely these strategies are working, and that is what we expected and incorporated in the data set. For cross comparison, we actually used melanoma as our starting point, and then the predictions were also incorporated for treating against other cancers. Again, you'll see in many of the situations we have higher numbers whenever we are applying these three strategies, and definitely gaining advantages from that aspect. Now, while comparing with the other three methods that I mentioned, and in some cases you'll see a few more here, when we had access to the code, or occasionally you might see one not listed there just because we had to use results from their publication rather than their code being accessible so that we can run it. IRnet is shown in the blue over here, and so many a times you will see it is either performing better than others or at par with the other data sets. And what you're seeing on this side here is the within cohort, which means the same cancer versus the same cancer prediction, training and testing. Third is the cross cohort, where mainly things were melanoma and then put on top of either cancer or gastric. The others are melanoma if they are not mentioned. Now, how do our users know what we are predicting is really important and is meaningful or not? And so, in order to bring that full circle, we did incorporate pathway enrichment aspects at the end. So what you see here is an overlap between top 30 ranked pathways for each one of these three different methods, which is the anti-PD-1, PD-L1, CTLA4. And what you'll see, a lot of them do land up hitting the common pathways in between, whereas some of them would have these unique pathways. Now, in order to compare this and get a better understanding from individual level to level, how does it perform in one patient versus the other? You can actually look at, so what you're seeing over here is the rectangles in this figure have the top 30 pathways, and you're pulling out three main top interactions from each one of them. The diamonds in the figures are showing some of the pathways which are known to be important for ICI treatments. And then some of the unique ones that you saw from the earlier Venn diagram are shown in an oval shape over here. So for each of these mechanisms here, which you see in the three different mechanisms, you'll see they are really pointing towards in different categories, color coded by KEGG in this figure over here, how the interactions reflect and how many of them are direct interactions between cancer related pathways, whereas some other times they will be pulling out some indirect pathways, but they have also been found to be really important. Now, lastly, we did want to make this available to our clinicians, to researchers who really are the non-informatics, non-CS background. And so in order to do that, we did set up a web portal, a web interface. It's publicly available if you go online at this URL. Our manuscript is currently under review. And when you actually go over here, the the website is really providing you options for not only looking at our own results, but really bringing your own data sets, doing your own training, doing your own predictions, and then getting all the way to the inference, similar to the figures that I showed earlier. And so when you're on the web portal, you could pick one or multiple and essentially run your prediction for whichever treatment type you need. You can use some of the publicly available data sets that we have provided, or rather bring your own data. So say if you have this information for hundreds and thousands of patients and really want to use this, you can bring it. You can also do this for other type of cancers. At this point, we are only focusing on the ones that we use for comparison. And then you can run your own predictions as well as view these predictions, so that what you're seeing here in the bar charts are the patients, multiple patients that we predicted, which were either responders or non-responders. You could simply click on a patient over here and it will land up telling you in terms of your pathways, either your pathways filtered by different weights. If you want to explore all of the potential pathways, they'll be color coded by the categories in KEGG. If you click on any individual pathway, it will land up giving you its topmost interactions with the other pathways, where you get to see the pathway names. And so this is really where we hope to really have more users directly go to the website, or essentially, if you have more data sets and are interested, we would love to expand our method for those cancers, and essentially also try to incorporate more of the ICI treatments. Again, all of this would not be possible without the hard work of my postdoc, Yushu Jiang, mentioned here. A few of the other students in the group who have also worked on these datasets or web portal development, as marked by yellow stars over here, and our collaborator, Dr. Dong Shu. Again, I'd just like to remind, if you're interested in one of our other works, G2P Deep, feel free to stop by in the workshop session tomorrow morning, and I'll be happy to take any questions for this.
Any questions for Trupti?
Yes.
So I know you said you used the KEGG library. I'm not certain if you would have thought to look at this or not, but I'd be kind of curious to understand if there's any impact on what the pathway library chosen was, or if there are any alterations to that library to see how they impacted prediction accuracy.
So not yeah.
Mind repeating the
Yeah,
question?
so the question is, if there are any limitations or impact that might come with the KEGG library, and in terms of how it may have impacted. So in terms of KEGG library, rather than library, I would say, when we are really using the pathway information for the networks, that information is coming from KEGG. And the reason for that is we really are looking at a graph sort of a network, and from KEGG we can really get an RDF network that is ready. Now, one limitation that we know from our work in this space is KEGS information is only, partially complete. It does not, especially in terms of cancer and immunology and pathways related to that. Reactome has a far bigger chunk. The only immediate application issue we had is Reactome had a kind of a hierarchical structure at this point, which is what we are really looking to get converted so that we can really fit our own model or expand our model so we can start working with reactome pathway, so that we hope to have both of those in future together and fill any gaps that might be there that we haven't addressed yet.